Hello, everyone, and welcome to today's webinar. This webinar is hosted by Jen and sponsored by Solvius. Thanks so much for joining us. Today's topic is strategies for determining critical quality attributes of nucleic acid therapeutics and their carriers. I'm Udwak Thomas, Senior Editor with Jen, and it's my pleasure to be your host for today's webinar. Let me start off by introducing today's speaker. Dr. Oliver Hocker is a scientist for biological mass spectrometry at Solvius. We'll get to his presentation shortly, but first, let's cover some background on today's topic. Determining the critical quality attributes of emerging nucleic acid therapeutics and their carrier systems poses a new challenge for biopharmaceutical companies. Low molecular weight nucleic acid therapeutics, such as antisense oligonucleotides or small interfering RNA, can be fully characterized using critical quality attributes like identity or purity with many different analytical tools. However, analyzing higher molecular weight molecules such as guide RNA, messenger RNA, or plasmid therapeutics remains difficult for most existing methods. In this webinar, we'll be discussing the advantages and limitations of current analytical techniques for determining the critical quality attributes of nucleic acid therapeutics, and we'll be doing so using examples from different therapy classes. Through this webinar, you'll gain insights into how physicochemical analysis of viral and lipid-based nanoparticles can provide useful characteristics of these complex systems. Today's presentation will be followed by a live Q&A session, so if you have a question for our speaker, you can enter it at any time in the Ask a Question panel on your screen. All right, let's get started.
Okay, hello and welcome to my presentation. My name is Oliver, and I'm a scientist from the company Solvius. And today I will be talking about the determination of critical quality attributes for nucleic acid therapeutics and their carriers. So first, some words about Solvius. Solvius is a contract research organization that supports the biopharma industry in bringing their products from the early research phase to the commercial phase. And Solvius is expert in comprehensive GMP analytical services for biopharmaceuticals and small molecules, also large molecules like ATMPs and cell and gene therapies. Solvius is located in several sites all over the world, with a headquarters in Kaiseraugst, close to Basel. So that's a very big pharmaceutical industry hub, but also in other sites such as France, Netherlands, and also we are expanding currently strongly in the United States. So the analytical methods always needed to co-evolve with the development of therapeutic molecules. So in the 1980s, most of the pharmaceuticals were small molecules, and they didn't require super sophisticated analytical tools to characterize their quality attributes. But over the decades, in the 2000s and 2010s, with protein therapeutics coming to the market with antibodies, antibody-drug conjugates in the 2010s, the analytical methods needed to be more sophisticated and more complex to assess the quality attributes of these more complicated molecules. And since the COVID pandemic in 2019, COVID-19, the mRNA technology really accelerated nucleic acid therapeutics, and also this forces the analytical tools to become better, and they're really at the limit nowadays. So today I will talk about nucleic acid therapeutics, such as mRNA that is formulated in lipid nanoparticles or adeno-associated viral vectors. So first I want to give you an overview about the therapeutic nucleic acids universe. So here just a limited overview, starting on the left side with short nucleic acids such as antisense oligonucleotides or microRNAs that have just 10 to 20 nucleotides in length. and a mass of maybe 5 kilodalton. Going to the right side, the plasmid that are in the several thousand to 10,000 nucleotide length with several megadalton molecular weight. And in between, we're having small interfering RNA that is double-stranded with conjugation. We have aptamers where the three-dimensional structure is really crucial, similar to proteins. Then we have guide RNA that is used for CRISPR-Cas for gene editing applications. And of course, mRNA, like in the COVID vaccine. And although at their core, they are just consisting out of 4 different nucleotides, so very basic composition, they can be much more complicated apart from their different size range. They can be modified in their phosphate backbone, also in the ribose unit, and also the nucleobase can be strongly modified. For example, a nucleobase or a nucleotide modification that led to the success of the mRNA vaccines was pseudouridine that increased the efficacy much stronger compared to the non-modified one. And in the middle below, we also have chemical conjugates, can be peptides, anything you like. And here's shown a triantennary GalNAc that promotes the uptake of the nucleic acid into liver cells, for example. And of course, on the lower right side, they can be also formulated in carrier systems, such as lipid nanoparticles that form a shell, a protective shell around, for example, mRNA or small interfering RNA against degradation, but also to improve uptake in the cells. Or of course, also adeno-associated viral vectors, so protein shells protecting single-stranded DNA. So regarding the critical quality attributes of nucleic acid therapeutics, they are still not as defined as we are used to for protein therapeutics. Some of the quality attributes are quite straightforward and clear, such as identity, meaning the right nucleic acid and nucleotide sequence. Also integrity, so that the molecule is intact and all nucleotides are present in the right order. Quantity and so on. But there are also some specific quality attributes, especially for the encapsulated ones on the left side, such as encapsulation efficiency or empty-full ratio, so meaning that the payload is also inside the LNP and not outside. Also the polydispersity and surface charge could be critical quality attributes. However, that's still not as clearly defined. Today I will be talking about the ones that are marked here in light blue, such as identity, integrity, encapsulation efficiency. So although all nucleic acids therapeutics at their core are just the 4 nucleotides, modified or not, from an analytical standpoint to determine a critical quality attribute, the size is very important. So we divided them here in 3 parts. On the left side, oligonucleotides such as aptamers, guide RNA, small interfering RNA that are rather small with 10 to 100 bases that can be perfectly assessed with mass spectrometry and chromatography. While the larger ones in the middle, the large single-stranded and large double-stranded on the right side, cannot be directly assessed with mass spectrometry due to their large size. So here we need to go different approaches by digestion, for example, or on the intact level, we need to use other techniques like capillary electrophoresis, or of course also sequencing and PCR. But let's start with some examples from the oligonucleotides, so the smaller ones on the left side. Here we see an example of a small interfering RNA that is analyzed with ion pair reversed phase coupled to mass spectrometry. And there are 2 different separations. So on the top one is the ion pair reversed phase and a denatured state. Normally the small interfering RNA is a double-strand RNA that has a triantennary GalNAc as a conjugate. And on the chromatography here, it denatures into the sense strand and the antisense strand, and also partially we see the double strand. In the lower one, in the helix, that's a more native state, where the double strand is still intact, so we see just one peak, the red trace. And both of them can be coupled to mass spectrometry. In this case, the denatured one, where we see three main peaks, double strand, sense, and antisense. And from all these three main peaks, we can also get mass spec information, where we get the isotopically resolved intact mass of double strand, sense, and antisense. Further, we can also fragment them in the mass spec to get information about the sequence to really confirm it's the right identity. Rather than these small peaks that you see around, they are degradation products or side products that can be also identified with mass spectrometry. So overall, we can cover a lot of quality attributes just with LC-MS technology here. If we go then to larger oligonucleotides, such as single guide RNA that are, for example, used for CRISPR-Cas technology, it's not 20 nucleotides, but 100 nucleotides long, so already much larger. And here we don't have so good chromatographic resolution. However, we can still connect it to mass spectrometry. And on the right side, the large peak is our main peak that should contain our wanted single guide RNA intact. And this we can also confirm, the black trace, 32.76 kilodalton, that's the correct mass. But also below this peak, we have a degradation product. That's a very typical oxidation, phosphosulfonate to phosphate conversion. Without the mass spectrometer, this would have been completely lost under the main peak if we would just use optical determination. So this shows that we can cover a lot of quality attributes with LC-MS. But if we go to the larger ones, such as mRNA or single-stranded DNA with several thousand nucleotides, we need to go in a different approach. And yeah, this is what I will go next to. If we want to analyze the integrity of mRNA, then capillary gel electrophoresis with fluorescent detection is the best way to go. The separation efficiency, the resolution is unmatched compared to chromatography. And here we extracted an mRNA out of an LNP system, similar to the COVID vaccine, with 4,300 bases. And we see for the intact mRNA more or less a sharp peak with some side peaks. We also stressed or stored the mRNA in the fridge at 2 to 8 degrees Celsius and for a year. And the pink trace shows the stressed sample. So we see the large single peak disappeared, and we have a lot of fragments. So if you compare this to RNA size standard, the gray trace from 500 to 6,000 nucleotide lengths, we see that we have a lot of fragments between 900 to 3,000 nucleotides. So this is a very good technique to assess the integrity and purity of mRNA. Of course, this doesn't say us that it's the correct identity, but in combination with Sanger sequencing or next generation sequencing that confirms each nucleotide has the correct order, this is a very good combination of both techniques. Apart from the integrity and the identity, there are also other properties of mRNA that are really important to assess. One is the 5' cap and the poly-A tail. So the 5 cap is very important for the stability, and also the poly-A tail on the 3' end is very important for the stability of the mRNA against degradation. And also it's required to attach the poly-A binding protein that allows the translation of the mRNA into the target protein. If these two things are not there, the complete drug doesn't work. Then apart from this, we can also digest the mRNA into smaller pieces, similar to a peptide mapping. So we cannot analyze the complete mRNA with mass spectrometry, but if we cut it into oligonucleotides with an enzyme, we can separate the individual pieces of mRNA and get mass spec information for each of these oligonucleotides and do a sequence mapping. Last but not least, we can also digest the mRNA into single nucleotides, where we should just get 4 peaks in the chromatography, the 4 base peaks, to confirm the identity, but also the ratio between these peaks or between these nucleotides. And in addition, we also see modifications of nucleotides. Now I want to go a bit more closer into the poly-A tail determination that we can do with LCMS. So the mRNA intact has its main sequence, here depicted in green, and also the poly-A-tail length that is important for the recognition and also the translation, as mentioned before, where we cannot analyze the poly-A-tail length on the intact molecule. So the poly-A-tail length is normally between 80 and 300 adenosines in a row. That's depending on the mRNA and the organism. And to assess this, we can do an RNase digestion that cuts the mRNA after each guanosine into fragments. But because it cuts after every G, the poly-A-tail is untouched and is released intact. Then to the mixture of our digested sample, we can add oligo-DT magnetic beads that are synthesized and contain a magnetic sphere where this oligo-DT is attached to. And this hybridizes in solution that we have a double strand, and with a magnet, we can take it out of the solution and have then a purified and concentrated poly-A-tail, detach it from the magnet, and then inject it on our chromatography. So on the lower left, you see then the chromatographic signal, a clear peak, where all different poly-A-tail lengths are combined, so we don't separate them in the chromatography. But we can get from this chromatographic peak also an MS signal. And here you see a distribution of all different poly-A-tail lengths. And each of these peaks has a mass, and with this we can recalculate the poly-A-tail lengths. So here in this case, from 117 to 130 adenosines length, and with intensity we know also the species that was the most abundant here in this case, 123 adenosines length. So with this, I want to go to the double-stranded nucleic acids, plasmid DNA, that can be used directly as a therapeutic, but also as a template for mRNA production. And the active relevant isoform is supercoiled, as shown on the upper left side. And then it's really twisted. But degradation can lead to strand breaks that can form open circular if one strand breaks, or the linear form if both strands breaks as impurities. And again, capillary gel electrophoresis is the most powerful technique here because it separates by size and by different shapes. And this example shows 2 different plasmids, one 4 kilo base, one 5 kilo base, and the 2 large peaks in the middle show the separation of these 2 different plasmids in one sample. But also on the left side, the linear forms are completely baseline separated, and on the right side, the open circular forms as well. So it's a very powerful technique that can identify these topoisoforms as impurities that almost no other technique can do. So with this, we are leaving the nucleic acids that are pure without any carrier, not encapsulated. And we are now coming to the LNP and AAV systems. And again, we have here 3 different groups of the encapsulated ones. One is on the left side, the molecular level, where we take the drug product, so the mRNA or DNA encapsulated, and we break the system open. We disassemble every compound that makes the LNP or the AAV into its subunits and identify and characterize them, starting with the lipid analysis for LNPs on the upper left, that the identity purity is the correct one. The same we can do for the AAV capsid proteins. Then, of course, we need to check that the payload is the correct one. that it's pure and that it's intact. And in addition, we can also check process-related impurities, such as residual solvent from the LNP production, where we need organic solvents to build the lipid nanoparticle, but also residual host cell DNA or other process-related impurities. Then in the middle, we have also the intact level. So in the drug product, we want to keep it as intact as possible, where we check the different size, charge, or polydispersity of the particles, where we use a lot of spectroscopic techniques such as dynamic light scattering, zeta potential measurement, Taylor dispersion, SEC-MALS, and so on. And of course, also the mentioned encapsulation efficiency or empty full ratio, where we can use microscopy, capillary electrophoresis, ultracentrifugation, and so on. Last but not least, you can also check the efficacy, like a potency assay, where we'll talk a bit about later. Well, let's start with the left side with the molecular level. So the lipid analysis is very crucial. So the lipids or the LNP systems are depending on the manufacturer, which size they want to have or which target tissue they want to approach with the LNP system. But in general, a good technique for this is HPLC-CAD, so charged aerosol detection, where we can separate all different kinds of lipid types and detect them with charged aerosol detection. That is a very universal detector. So on the upper left side, you see a chromatogram, where in the lower retention time range, you see the pegylated lipids that are rather broad peaks, because these pegylated lipids have attached to their core structure polyethylene glycol that is very polydisperse, and we're separating here these different PEG chains already. Then they contain mostly cholesterol, and then in the upper retention time range, you can see the separation of cationic lipids or ionizable lipids. And these are very crucial lipids for lipid nanoparticles. Without them, it would not work as it does, because at lower pH value, they are positively charged. So they can, in the manufacturing, can bind to the phosphate backbone of the mRNA and complex with it. And in higher pH values, they form then this lipid nanoparticle. Then when these LNPs arrive to the target cell and go into the cell, where the mRNA is then released, the pH value is again lower, around pH 5, where the lipids get again cationic and release the mRNA. So here these chromatograms show 4 different LNP systems where the LNP was disrupted, and the identity and also the quantity of each lipid was determined. So the lower left side, you can see we have a good way to quantify them. In a similar way to the lipid analysis, we can also determine the protein shell of the adeno-associated viral vectors. So for the AAVs, the single-stranded DNA in the center is surrounded by a protein shell. And this protein shell consists out of 3 different proteins. They are VP1, VP2, VP3. And we can denature this capsid shell, that the proteins are not forming this shell but are free in solution. And we can determine their ratio. That is really important. That needs to be around 10 to 1 to 1 for VP1, VP2, VP3, as shown in the lower left electropherogram with capillary gel electrophoresis. Here are 3 different serotypes as a comparison. Apart from this, we can also apply classical molecular weight analysis with LCMS. On the upper right, the separation here is not as good of VP1 to 3. But for each of these protein peaks, we can determine the average mass, and also we see the glycosylation pattern that is really characteristic for the different serotypes. And last but not least, we can also do a classical peptide mapping where we digest the protein into smaller peptides to really confirm the correct amino acid sequence of each of these proteins, and also see modifications and post-translational modifications of the peptides and where this modification is located to really deeply characterize this protein. So with this, we are leaving the molecular level, so the subunit analysis, so to say, of one of these particles, and we are going more on the intact level. And what is really visually and nice to see is microscopy images. So here on the left side, we see cryo-EM image of lipid nanoparticles, similar to the COVID vaccine. And we see the image is quite heterogeneous. So we see spheres of different sizes that are the lipid nanoparticles. We see single ones, we see aggregates. And we see also some that look like empty, some that are filled. And we even see mRNA that is outside of the lipid nanoparticle, so not protected at all. On the other side, we see TEM, so transmission electron microscopy image of AAVs, so viral particles, that look from the size much more homogeneous, look like tennis balls, and some are whitish inside and some are black. This is due to staining. So the ones that have a black center, they are empty. So the encapsulation process is also not perfect for virus production. And the ones that are white inside, they are filled with a single-stranded DNA. But although they look nice, these images, they cannot be evaluated in an efficient way, not automated. So we need other techniques to assess the empty-full ratio and other properties that I cannot now go too much in detail, but I want to show you a technique that is very suited for empty-full ratio of AAVs. That is analytical ultracentrifugation, or AUC. That in principle is a spinning cuvette, like a centrifuge, but that has a detection window where we can have an optical detection, UV detection with 260 and 280 nanometers. And in this cuvette, while it's rotating, particles of different mass are sedimenting in a different rate. And this can be measured over the optical detection. So the ones that are correctly filled are represented by the large peak more or less in the middle, where we see this is like the highest proportion of our sample. Then on the left side, the second highest peak is the empty capsid. So that's a capsid that has no filling inside. And then in between these both, the small peak is a partial filled capsid. So it has a payload. It can be inside a fragment of a DNA or some other filling, unknown filling. And on the right side, we also have aggregates or overfilled capsids. So capsids that have more filling than it should have. And yeah, in Solvias, we're having this also running on a GMP. That is something very unique in our business. And that's like the gold standard for determination of empty-full ratio for AAVs. Then in analogy to the AAVs, for the LNPs, we can also do this. So lipid nanoparticles are also not perfect in encapsulating the mRNA. Ideally, it should be inside the lipid nanoparticle to be protected, but there's also always mRNA outside of the LNP. Normally, this is done with a ribogreen assay, but this is not flawless and has some disadvantages. So we were developing another approach that is quite similar, but involves capillary electrophoresis. So we add to our sample a fluorescent dye that in contact with mRNA starts to emit fluorescent light. And this labeled sample we inject then in our capillary electrophoresis system. And only the mRNA that is outside is migrating to the detector. So here the blue trace, we see the free mRNA that was not inside an LNP. And then in the second step, we disrupt the LNP with surfactants or organic solvent. And then also the mRNA that is inside or was inside the LNP is now released, gets labeled, and can be measured again. That is here the pink trace, where we see the total mRNA, and with this we can then calculate the encapsulation efficiency very easy. So with this, we come then to the last box, the right one, the efficacy, the potency assay. And this potency assay is very demanding. It requires a lot of expertise that is working in combination that we at Solvias can provide. So in a cell-based potency assay, you use your nucleic acid therapeutic, that is as you would give it to a patient, but you give it to a cell. So the nucleic acid therapeutic is then entering in the cell, a transfection. And the cell you keep in a cell culture, grow the cells, and keep them basically alive. And then within the cell, due to the therapeutic, the effect happens, and the target protein is expressed, or a target protein is reduced. And these cells you can then harvest, do cell lysis, and extract the target protein over a specific sample preparation. Then also identify the protein with various analytical techniques of your choice. Can be capillary electrophoresis or ELISA, SPR chromatography, LCMS, and so on, to quantify it. And then with this data that you can interpret, you can calculate a response related to a dose. So you know how much of your drug product you gave to the cell and what is the response, so you can determine a potency of your drug. So, and with this, I want to end my presentation. Thank you very much that you were here today and took the time to listen to my talk. And yeah, I'm happy to answer your questions in the following question and answer session. Thank you.
I'd like to thank our speaker for an informative and interesting presentation. We're going to begin the live Q&A session in just a few moments, so please submit your questions if you haven't done so already. Before we start the Q&A, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please do keep an eye on your email, and please do share the link with your colleagues who might not have been able to tune in today. All right, just give us a couple of seconds. And welcome everyone to the Q&A session for today's webinar. Thank you all so much for being here. And thank you for listening to the presentation. Oliver is here and ready to take your questions. So I just would like to let you know, if you haven't already asked your question and you do have one, please go ahead and enter your question in the Ask a Question panel on your screen. We're going to be taking questions for several minutes, so please go ahead and enter your questions now. Oliver, thank you so much for that presentation. I'm going to jump right into the questions we have. First question I have for you. I'm interested in AUC and the GMP qualification at Solvias. As far as I know, there's no AUC software which can be qualified. Can you explain in a little bit more detail about how you do this at Solvias?
Yes, for sure. So in Solvias, we use analytical ultracentrifugation for many characterization projects, but customers more and more requested AUC also for release testing. So if you have a drug and you want to release it to the market, to the patients, you need release testing that is GMP qualified. So since there's no software available that can be GMP qualified, that is commercial, some of my colleagues that have interest in IT and programming together with our IT department decided to write their own software in-house in Solvias that controls the entire data flow. So the data are evaluated normally with a software that is called SetFit. And this is used more or less by everyone that is doing analytical centrifugation, but it's not GMP compliant. You can delete data and manipulate whatever you want. But this additional software that was written in-house is directly downloading the data from the machine after the data are acquired, saving them on the server that is backed up, and adding an audit trail to every task, every step that was done in the instrument, and also to the set-fit software, where the analyst is setting any parameters, doing data evaluation. So everything gets a timestamp, who did it, what did he do, which settings. And like this, we can trace everything back for decades, actually. And also other companies, they offer analytical authentication under GMP, but normally they're doing this with the 4i principle. So it's just another person watching the analyst performing the task. And yeah, that's not so up to date anymore. So this software solution is much more better.
Excellent. Excellent. Thank you. All right, I have another question here for you. Regarding the LC method for the nucleotide analysis of mRNA, what type of chromatographic column do you use?
So we tested several columns from ion exchange chromatography and helic columns, and they all work and they all separate the nucleotides. But our most favorite column was reversed phase phase, and there are traditional Waters HSST3 column, a HPLC column, where we can separate all nucleotides, also all DNA nucleotides, so deoxynucleotides or oxynucleotides. So we can separate DNA nucleotides from RNA nucleotides, also nucleotides from nucleosides. And this is a very high performance method. Under 15 minutes, we can separate all of these. Also separate modifications, methylation, and so on. And with the reversed phase, of course, you have the advantage that you are MS compatible. So you have also, in addition to the chromatography, the MS signal, where you can also do MS experiments to really confirm what kind of nucleotide or nucleoside you have there.
Excellent, excellent. And thank you to our audience for those questions coming in. Please do keep them coming. We're taking questions for the next several minutes. So I have this question from our audience. Do you have experience with any other nucleic acid species? So for example, single-strand DNA, circular RNA/DNA.
Single-stranded DNA, yes, especially from the AAVs. There we have also experience in purity analysis, identity analysis. With circular RNA, I'm not so sure if we had this in-house. I didn't work still with this, but of course, I heard that this is a thing at the moment.
Right. Next question I have, can single-strand sequences of DNA be encapsulated into LNPs?
I'm sure it can. I never heard about this, but I don't know why it would not be able to be encapsulated into LNPs. The thing is, really long DNA strands, to my knowledge, there are not so many therapeutic applications. But yeah, from the principle, it should be possible. Also, DNA is normally much more stable than RNA. So the purpose, of course, of the LNP, as you probably know, is to protect the RNA against degradation. There are enzymes everywhere that degrade RNA into smaller pieces, RNases. DNA is normally much more stable. But of course, maybe to bring these DNA to a certain cell or target, maybe the LNP could be also very useful. I think this should be possible.
Excellent, excellent. Next audience question I have, what are the advantages of CZE lift to compare with ribogreen for RNA encapsulation efficiency?
So if you add the ribogreen to your sample, it's observed that the ribogreen also enters or can enter quite quick into the LNP particle and also stain the payload, so the RNA inside. So the signal that you observe after a while when you measure the fluorescent output can be a bit biased because you are also measuring the fluorescent light that comes from the RNA that is inside the LNP. So if you use the separation technique, then it cannot be biased because you are separating the free RNA from the LNP definitely, and you measure only the response from the free RNA that is outside. So this is one of the biggest advantages of this approach.
Excellent. Excellent. Thank you. All right, I'd like to go to this question. Where do you see the applicability and use of oligomapping by mass spectrometry?
So oligomapping is a quite new approach. It's was maybe really developed or published with the release of the COVID-19 vaccines. So it didn't really arrive to QC labs, I would say. It's still a bit in the research phase. But the advantage of it would be similar to a peptide mapping for proteins. So you confirm the sequence, so similar to next generation sequencing, Sanger sequencing. So with this, you could confirm the sequence, so the identity. You can confirm that all nucleotides are in the correct order, that also the sequence is complete. And you can also identify modifications. So if you break the complete mRNA into pieces, you separate it over the chromatography, you get these peaks of each fragment, and you can analyze them in the mass spec. You can also see parts of the mRNA that may be modified, so they have then a different mass that you maybe would not see with other techniques. So that's a really big advantage of oligomapping, because you also cover many quality attributes at once in principle. So the problem with oligomapping is that it's rather new. So there are not so many software solutions. For peptide mapping, there are a lot of softwares. It's a really standard method nowadays. For oligomapping, there are still not so many software solutions to handle the data that you create. Also, there are not so many enzymes to digest the mRNA. For peptide mapping, there are several enzymes that work perfectly well and that have not so many miscleavages, and everything is known for proteins. For mRNA, it's not like this. It's still very young, and there are not so many specific enzymes. So for example, RNase T1 is cutting after every guanosine, but because we have only 4 nucleotides, there are so many guanosines that you cut your protein in very small pieces. So then these small pieces are not so specific anymore, not so unique. So then to assign these bits to the part of the sequence to cover the whole mRNA is really difficult. So normally there are also not so high sequence coverages like in protein peptide mapping, for example. But I think it's strongly worked on. Many companies like Pfizer and Moderna were publishing a lot of literature publication in this field. So I think it will grow much faster, and maybe in some years that's a typical quality control technique that we will apply.
Excellent, excellent. Thank you to our audience for those questions. I see them coming in. I'm going to take this one. What fluorescent dye do you use for encapsulation efficiency using CZ-ELIF?
We are using mostly cybergreen. So with cybergreen, we also know that it definitely penetrates into the LNP, but because we are separating it, so we are separating the free mRNA from the encapsulated one, we don't care that it's penetrating the LNP, because we can really separate the encapsulated from the free one. But yeah, we are using cybergreen.
Excellent, excellent. And then this next question is a two-part question. So what strategy should be used if we change the method of analysis for a particular CQA during clinical development? Can we justify it, or do we need to present some data?
I'm not so sure if I understand the question.
Yeah. If the audience member asks this question, if you don't mind re-asking the question, we'll come back to it and we'll try to answer it live if we can.
Yeah, of course. We will go into the question, and after the webinar, I will get in contact with you.
Absolutely, absolutely. All right, let me ask this one, only because I think the next question, there are two questions that I may be asking the same thing regarding RNases, RNase enzymes. Can you elaborate on the types of RNase enzymes for digestion? Are there different ones for poly A tail characterization and a full oligonucleotide mapping?
Yes, so as I said before, there are not so many specific RNases. So the RNase T1, for example, that I mentioned before, is cutting after each guanosine. And for this, it makes very small pieces. And this is okay for the poly A tail determination, because the poly A tail, of course, consists only of adenosine. And so the complete main part of the mRNA gets chopped into small pieces, and the poly A tail remains intact. So poly A tail has maybe 80 to 150 to 200 nucleotides. So RNase T1 is suited for poly A tail determination. So then with magnetic beads, we can take out the poly A tail from the sample. But for an oligomap, we want to have specific and like fragments from the mRNA that we can assign to parts of the sequence. So they should be a bit larger. And other RNases that came up in the last time in the recent publications are RNase H, or also RNase 4. And RNase H, for example, RNase 4, cuts after uridine. Well, not after every uridine, but I think in combination with U and an A, or U and G. So this limits already the cutting sites, because the chance that there's an UA and a UG is not so high like for the RNase T1, where it cuts after every G. So then with this, we get much larger fragments that can be better assigned to any parts of the sequence. There are also other approaches like hybridization, so that you add to your sample like probes. You synthesize specific probes that hybridize with a part of the mRNA specific sections. And then you can increase the portfolio of RNases that you can use that cut specifically double-stranded RNA. So if you hybridize your single-stranded mRNA on some parts with a short oligonucleotide, you have a section of a double-stranded RNA, and the enzyme can cut very specific.
Good. Excellent. Next question I have for you: how strong is the influence of ion pairing agent concentration on the mass spectrometry signal intensity, but also on the separation efficiency? And then the second part of that question: are there sweet spots which combine good chromatographic resolution with sensitivity?
Yes. So first of all, in reverse phase chromatography, you need the ion pairing agent that you get retention for nucleic acids, for oligonucleotides or mRNA. So normally the nucleic acid is very hydrophilic because of the phosphate backbone, has a lot of negative charges. And in reverse phase, normally you separate by hydrophobicity. And nucleic acids, they normally elute just in the dead volume because they don't interact with the stationary phase. So for this, in ion pair chromatography, you add an ion pair agent. So this ion pair agent is normally quaternary amines, so with a cationic head that is interacting and bonding with the negative phosphate backbone of the nucleic acid. And then it has a hydrophobic tail, and this hydrophobic tail then interacts with the stationary phase of the, for example, C18 material. So this ion pair agent is like a glue between the nucleic acid and the stationary phase. And the mass spectrometry, so electrospray ionization, normally is really strongly disturbed by these ion pairing agents. So in the mass spectrum, you will see, if you use triethylamine as an ion pair agent, you will see millions counts of triethylamine, and it's also taking away charges from your actual analytes, so you have a reduced sensitivity. It's really not so good for your mass spectrometry sensitivity. So for this, you should reduce the ion pair concentration to have better sensitivity. But the more you have in your eluents, the better is your chromatographic separation, the higher is the retention of your analytes on the column. So of course, then there's like a compromise ratio. But then it was found out that you can add fluorinated alcohols like hexafluoroisopropanol, for example, that is boosting the interaction of the ion pair agent with the oligonucleotide. So you can reduce the concentration of your ion pair agent by adding these fluorinated alcohols. And also these fluorinated alcohols, they have a good influence on the electrospray process, so they are boosting sensitivity. So with these additives, you can supercharge and balance out the negative effects of the ion pairing agent. And of course, then there are different ion pairing agents. Triethylamine is one of the most universal ones that you can start with, but there are also different ones like butylamine or hexylamine or dibutylamine or something like this. And actually, the longer the hydrophobic tail is, the better it's suited for very long oligonucleotides or for RNA, mRNA, and so on. So with this, you can play a lot.
Excellent. And just a last call to our audience for questions. We have a few more moments, so if you do have a question, please do go ahead and enter that question. We're taking questions for a few more minutes. Let me ask you this one. Can you further explain why there are limitations using mass spectrometry for intact analysis of nucleic acids, such as mRNA? You said that mRNA can be analyzed by MS only in the digested form?
Yes, that's true. So we test this also ourselves, and if you have a 1,500 nucleotide RNA and you try to analyze it with a mass spec, the MS spectrum is just not usable. It's a very broad distribution of a lot of charges, a very complex profile that cannot be deconvoluted. Also in the literature, you don't find any publication with mRNA or RNA analysis above 1,000 nucleotides. And this is because first, the mRNA or the large nucleic acids, they carry then so many charges that the convoluted MS spectrum is so dense that the resolution normally is not enough, and you have so many different charge states that you cannot deconvolute anymore in the end. And also already with proteins, with antibodies, with 150 kilodalton, 200 kilodalton, 300 kilodalton, these are already quite large, but mass spectrometers can handle this, can cope with this. But you need to see that mRNA, like the COVID vaccine, is already in various megadalton. And this is just a size of a molecule where the MS hardware is not designed to deliver the ionized molecule through the ion guide. It's just not really made for this. So, but there's another technique that came up in the last decades, more or less. It's still not ready. It's still in the research phase, but it's coming more and more to the market. Also pushed by like virus particles or plasmids and also mRNA, because there are just not enough techniques to analyze these big molecules. And this is charge detection mass spectrometry. And this is a kind of mass spectrometry that measures not all ions that come in at once, where you get this different distribution of a molecule that has 30 charges, 40 charges, 50 charges with different adducts that make up this very complex spectrum. But it takes one ion of this and analyzes the charge of the ion and also the mass, and then takes another ion, measures the charge and also the mass. So it's a single particle detection method. And with this, you can really accurately determine the mass of very large molecules, such as complete virus particles and plasmids. However, at the moment, this technique is very slow. So to get good data of virus particles, like an AAV, you need to measure for hours one sample. So it's at the moment not able to be coupled with chromatography, for example. And it's at the moment still not practicable. It's you need a lot of optimization. But I also expect that in the future, the technology will get much better, faster. And Thermo, for example, brought out an instrument. Also Waters is working on it, but they're still a bit in the research phase.
Excellent, excellent. I think we have time to take one more question. So let me ask you this. Which critical quality attributes are the most challenging to assess with analytical tools these days?
Yeah, I think this, we heard on already a lot today. So it's the size mostly, because they're just so big. And I think also with upcoming drugs that are more conjugated, there are nucleic acids with protein conjugates formulated in lipid nanoparticles or other unbelievable combinations. So to analyze these large constructs is really difficult, because there's no analytical technique that can fully analyze this one thing completely. So we need to deconstruct the molecules into its subparts and combine so many different analytical techniques, and also later combine all these data together to make sense out of it and to really assess, is this drug now safe? Has it a good quality or not? And I think that's the largest challenge nowadays.
Excellent. Excellent. Oliver, thank you so much once again for your presentation and for taking time to answer questions. That is all the time we have for today's webinar. And so to our audience, just so you know, this webinar will be available to view on demand within the next 24 to 48 hours. So you can come back and watch the entire webinar from start to finish. And you can also share it with your colleagues who might not have been able to tune in today. So just keep an eye on your email. You should get that link within the next 24 to 48 hours. Once again, Oliver, many thanks for being here. Thank you so much for your presentation.
Yeah, thank you for having me, and thank you for watching. Thanks.
Absolutely. I'd also like to thank our sponsor, Solvius. Thank you so much for sponsoring today's event. And I'd also like to shout out the Gen Multimedia Group for hosting. So thank you, David Mosley and Reynolds Gutierrez. And last but not least, thank you to our audience for being here. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.